So good morning, good afternoon, good evening, everyone. Welcome to the virtual European MedDRA User Group meeting, which will address MedDRA, vaccine vigilance, and COVID-19. I will chair this session. This is me, Martin Menke, Liz Thomas, which you already have heard, and Brian O'Hare from the MSSO will provide the technical support. And we'll start with some housekeeping. So we want this to be interactive, so you can place your questions. On this first slide, you do see kind of how to do this. So you just pop out the control panel, which is located on the upper right of your screen. You type in your question in the question box, and you send it off by clicking on the send button. We will have a short questions and answers session after each of the presentations. And at the end, we will try our best to answer all of them, but we provide no guarantees. It's dependent on what time permits. And with that, some more frequently asked questions. Are you going to be able to get the copy of the slides? Well, yes, and it is recorded, as Liz just said. You'll find them on the MedDRA.org user group, but actually the PDFs of the presentations are also already attached to this webinar, and you'll find it under handouts. It's kind of the second from the bottom of the control panel. So there you will find the PDFs. And I'll present, and my role is actually as chair of the European Industry MedDRA User Group Steering Committee. And here you see the whole lot of us. So there's a larger group behind this, and we're also very thankful for the MSSO to provide input and support us and provide content as well as usual. So with that, a heartily welcome on behalf of the European Industry MedDRA User Group Steering Committee. And generally, we start this with some questions we're having to you. And this will be done via Liz and Brian via the respective functionality in this product here.
Okay.
Well, thank you, Martin. So just to get a sense of who's with us today, we had a couple polling questions and also some questions about the MSSO's face-to-face training program that we'd like you to participate in. So what I'll do is I'll ask Liz to put up the first question, and this is, you'll be able to respond to these questions directly into the GoToWebinar software. So Liz, if I can ask you to launch the first question. So from what region are you attending this webinar? You've got a number of choices there, so if I could have everyone respond to that. And we'll post the results here in just a moment. Okay, I'll just give a few more seconds for people to answer that question.
Okay.
Okay, Liz, why don't you go ahead and post the results. Oops. There we go. Okay. All right, very good. So we have people from different parts of the world, mostly from Europe as we expected, but also North America and other parts of the world. It's very good. So let's go ahead and go for the next question, Liz. Okay. Next question is, what type of organization do you work for? So are you a CRO? Do you work in a regulatory authority? Are you a device organization, or are you a pharmaceutical company manufacturer distributor? I'll just give a few more seconds. Okay, Liz, why don't you go ahead and post the results? Okay, very good. So we've got half of our users with us today are with a pharma company, manufacturer, distributor. Excellent. Okay. Next quick question here. And what is the role in your organization? Are you focused on pharmacovigilance, or are you on the research side, clinical operations, coding, or are you more focused on statistics or informatics? Okay, just a couple more seconds here. Okay, let's go ahead and show the results. Okay, very good. So we've got majority of folks in drug safety, pharmacovigilance focus, and the others spread amongst coding, statistics, etc. Okay, let's go for the fourth question. These final two questions have to do with our the MSSO's face-to-face training program. And some of you may have taken these in-person classes. So we're trying to poll our user base to determine how soon the MSSO should resume face-to-face training classes, given the current COVID situation. This is just helps us get a sense of when we can begin to focus and prepare ourselves to provide this service again. So you can see the choices there. There's last 2 quarters of 2021, the first quarter of 2022, or you're just not sure. So I'll give you just one more moment to respond to that. Okay, Liz, why don't you go ahead and provide the results? Okay, so not sure. Okay, well, thank you for that feedback. I think the majority of folks that have answered this question have selected not sure, so that's something that we want to continue to monitor. And then the final question related to this, if you can post that, Liz, is, I'm assuming this is after the COVID situation is over, would you be willing to travel or attend face-to-face mentor training? So the possible answers there are when we restart our face-to-face training program, or do you prefer the virtual offerings that we've been providing during the COVID situation? Are you on the fence, maybe, or you're just not sure? So I'll give just a few more seconds to respond. Okay, Liz, go ahead. Okay, so again, a lot of people are on the fence here. So thank you for that feedback. That ends our polling for now. So Martin, I'll turn things over to you.
Good, thank you, Brian. Thank you, Liz, for this. It's always important to know these things, to develop our services, to service you as required. And with that, we go into our agenda, actually. So the agenda is on MedDRA vaccine vigilance and COVID-19. We will start with the update from Judy on MedDRA, followed by a very interesting talk by Barbara Law from the Brighton Collaboration, and from WHO, Anna Baumgarten, and the final talk will be from Kendall Harrison on MedDRA, all focusing on what they are actually doing concerning vaccine vigilance and COVID-19. And the last part will be then the open house question and answer session. So after each session, there will be some time for the questions, and the final open session. But with this, I will hand over to Judy, who will talk or will provide a MedDRA update, and she will and many of you will already know her because she's the Chief Medical Officer of the MSSO. So with that, Judy, the floor is yours.
Thank you very much, Martin. Just switch over to my slides. Okay, and hopefully you can see that. Well, welcome to all, and thank you for joining us today. I hope that you're all keeping well. On behalf of my MSSO colleagues, I'd like to say how pleased we are to be able to host this webinar with the MedDRA user group from Europe. And we're really interested in looking forward to the presentations from our expert panel today on the subject of COVID-19 vaccine surveillance. This is the second year that we've had to host this virtual meeting due to the pandemic. And I'm sure many of us miss meeting our friends and colleagues in a nice European location and for a full day face-to-face meeting. However, as we saw from the polling in the beginning, there are some advantages to this virtual format in that we can reach many more people in a wide geographic distribution. And also this webinar is being recorded, so we can reach a wide audience with this. So I will present an update on MedDRA and MSSO activities. Now, first off, the buzzword today in pharmacovigilance is terminology interoperability. So I'll be talking about two mapping initiatives, one with Snomed CT and one with ICD. And then three new topics. We have a new SMQ in development. I'd like to introduce you to some new MSSO staff and also talk about some new training offerings. So first off, the Snomed CT MedDRA Maps. This provides an overview of where we are with the development of these Snomed CT MedDRA Maps. The headline here is the first production release of these maps will be in just 10 days' time on the 30th of April. Now, we've talked a lot about the development of these maps in the past, but they were developed as part of a collaboration of regulators and industry partners through the WebRadar 2 project, which was focusing on developing interoperability between terminologies and using mobile application technology to be able to facilitate the transfer of clinical information between regulatory systems, which use MedDRA, and electronic healthcare records, which use Snomed CT. So a mapping between those two terminologies is fundamental to achieving that goal. As part of the project, which ended last year, we developed a bi-directional map of a subset of MedDRA and Snomed CT terms. So it's not all of MedDRA or all of Snomed CT. The first production release in the end of this month will use the January 2021 version of Snomed CT and version 23.1 of MedDRA from last September. It does include some mappings of relevant COVID-19 terms. Now, both Snomed users and MedDRA users are able to access these maps, and you're also able to contribute to the development of them because a key part of this project is the sustainability of these maps in the future. So I'll describe how you're able to contribute to the development by using our new online MapCR tool to make changes to the maps. And we intend to update these maps each April. Now, what do the maps look like? Well, they're two independent maps, one in the direction from MedDRA to Snomed CT, and one in the direction from Snomed CT back to MedDRA. And what we're aiming for here is matching exact concepts within each terminology so we can translate easily from one to the other. Now, the MedDRA to Snomed CT map is slightly bigger than the Snomed CT to MedDRA map. And that is because there's more granularity because we're mapping at the MedDRA LLT level to the corresponding concepts in Snomed CT at the FSM level. So this is a fully specified name, and that's more like a MedDRA PT. So you'll see, for example, in the top 3 LLTs there in column B, they're all lexical variants for an abdominal aortic aneurysm, expressed in different ways in the LLT and MedDRA. However, in the Snomed CT terminology, you'll see that they all map to a single FSN. So the MedDRA to Snomed map comprises about 6,500 terms. In the reverse direction, Snomed CT to MedDRA, this is made up of about 3,700 terms. And here you'll see for the abdominal aortic aneurysm in Snomed CT in column B, that just is a one-to-one map back to the corresponding MedDRA LLT abdominal aortic aneurysm, which is an exact word-for-word match. So the maps are actually distributed for MedDRA users in this Excel spreadsheet format. It's a tab for each of the 2 independent maps in a single spreadsheet. Snomed CT users use a different HTML-based format, which Snomed CT users can access from Snomed International. So for us MedDRA users, how do we access the maps? Well, on the 30th of April, they will be available on the downloads page, which is shown here. You will need your MedDRA user ID and password to be able to access these maps, just the same way that you access the releases of MedDRA. So the map files will actually live here in the spreadsheet format. There's also supporting documentation to aid users in understanding how to use and interpret the maps. On the tools page, which is under the how to use section on our MedDRA website, you'll see a cog for mapping. And that if you click on that, it'll take you to a number of documents, including mapping conventions, the release notes, and a link to the MapCR user guide and the application itself. So we've yet to post these on the website, but you will see them on the 30th of April. So let's talk a little bit about this MapCR tool. The MSSO developed this tool to enable either MedDRA users or Snomed users to be able to access the maps. If you're familiar with our WebCR tool for submitting online change requests to MedDRA, it has a very similar look and feel. So you have to be able to log into it, and then you can request terms to be added to the map, either starting from Snomed CT to MedDRA, and then we will automatically map back in the reverse direction, or you can submit MedDRA terms that you want to be mapped to Snomed CT and then back in the reverse direction. You can also request a revision to an existing map if you think it is incorrect. And you can also, similar to WebCR, search already submitted requests for changes from other users, and it's all anonymized. Now, similar to our requests for new terms to be added to MedDRA, if we are going to add new terms to be mapped, we need to have a clear use case. Why do you need this? It's fundamental to the change request process. So you have to provide a justification either based on a need from a regulatory, clinical, PV, EPI perspective, but you have to say why you need us to do that work to implement that in the maps. So a real world requirement. And here are some of the use cases behind those types of issues. For example, if you have a black box warning on your product, which is coded in MedDRA, and you want to make those particular adverse events or contraindications, warnings visible in the electronic health record, we can take that MedDRA coded data, convert it to its equivalent in Snomed CT, and so a prescribing physician could be able to see those warnings in the language in the electronic health record using Snomed CT. Another use case in the other direction that we might be interested in is accessing large healthcare databases to look for adverse event monitoring. This is particularly relevant now in the context of COVID-19 vaccines. So we might want to look at Snomed coded data and electronic health records to look for any relevant adverse events, convert them into MedDRA for the purposes of recording and analysis. Another use case that we've heard about is actually looking at Snomed coded data in electronic health records to look at patients' medical history and determine whether they might be eligible for inclusion in a clinical trial, for example. And we can again convert that data back to MedDRA for our work in industry or regulatory databases. Now, the next slide shows just a prototype of how the MapCR entry tool will appear. You're required to enter your name and email address because we will be communicating with you, as we do with WebCR, on the disposition of your request. And we can get back to you if we need any further clarification and tell you what the final outcome was. So you can start from either terminology as the source, either starting from a set of MedDRA terms that you want to be mapped to Snomed, or vice versa. There's also a field to include your rationale for why you need this, these terms to be mapped. And if you wish, you can even propose some target codes that you would want this MedDRA term mapped to this Snomed term. So it gives us some help as a starting point where you're thinking. So there will be a detailed user guide to accompany this online tool. So it will help us to maintain these maps going forward. So our next topic is ICD to MedDRA mappings. And I know there's been a lot of interest in the user community over the years to have a standardized mapping from ICD-10 to MedDRA to be able to look at claims databases and registry data, for example, and convert that information to MedDRA for the purposes of adverse event reporting and other activities. So we're pleased to say that the MedDRA Management Committee has supported the MSSO to pursue the development of an ICD-10 to MedDRA mapping. Work is beginning on that. It will be from the core international version of ICD-10 to MedDRA, just in that one direction. And looking to the future, the MedDRA Management Committee has also supported exploration in conjunction with WHO to develop and maintain mappings between ICD-11 and MedDRA. And these would be bi-directional in nature, similar to Snomed and MedDRA Maps. We're just starting this project, so any more details will come to you as we work them out. But for the moment, I can't really say much about our projected timelines or the formats that the maps will be in. But just to let you know that this effort is underway, and I'm sure it will be welcomed by our user community. So next on to some new topics. And we are developing a new SMQ for sexual dysfunction. This has been approved to go into development by the MedDRA Management Committee. And as part of our process, the MSSO reviewed the initial request with the points to consider working group. And that's where we get valuable feedback from regulators and industry people about the need to develop this as a query. We found that the terms that represent this condition are scattered across multiple SOCs. So really an SMQ is an ideal way to be able to address this particular query. And it's important because it forms class labeling for many products on a global basis. Antidepressants such as the SSRIs and SNRIs are known to be associated with sexual dysfunction, not only while taking the product, but they can also be associated with the persistence of this condition even after discontinuation of the product. Also, oral contraceptives, finasteride and levodopa have class labeling for sexual dysfunction. Now, in discussions, we decided to develop this SMQ without the need for testing. Typically, the MSSO will set up inclusion-exclusion criteria, a draft term list, and then actually go and test it on regulatory and industry databases to make sure that the terms that we provide reliably retrieve relevant cases. But in this case, we felt that the need for testing was not there because the search terms have already effectively been validated in class labeling. We know that these types of terms that appear in the labeling reliably retrieve relevant cases. So we're able to do this in a slightly shorter fashion without the need for testing. The MSSO has drawn up some draft documentation and a draft term list that we'll be reviewing over the next couple of months with international regulatory and industry experts. We will then take the finalized documentation and term list to the MedDRA Management Committee, and we hope that they will approve this SNQ to go into production for 24.1 if we're able to meet all of our timelines. Next new topic, a warm welcome to our new MSSO staff. You might be seeing them coming up on future webinars. First of all, Dr. Yelena Obenchina. She's based in Moscow, Russia, and she will be providing local support through the help desk and also training to Russian speakers in the Russian Federation and other countries in the Eurasian Economic Area. And she's already got her feet wet with presenting a webinar in Russian. We're delighted to have Elena as part of the team because she has a long association with MedDRA. In fact, she led the team that developed the Russian translation of MedDRA, so she's very well acquainted with the terminology. Our next new staff member to welcome is Nicole Fonorato. She will be working on mapping initiatives, including the continued maintenance of the Snomed CT mapping, and also on the development of ICD mappings. And she will be also conducting US-based training. But you'll probably see her on webinars across the globe as well. Nicole has a very long history of MedDRA, going right back to even the pre-release of MedDRA when she was an intern. Most recently, she's been Associate Director of Medical Coding at Celgene, and she's based in New Jersey, US. Our last topic is new training offerings. This grid shows how much we've expanded and listened to you as our users to expand our offerings over the course of last year. There was an explosion of new courses added in the later part of this year. On the left-hand side, we've already talked about our face-to-face courses. Currently, the only location in the world where we're conducting face-to-face courses right now is in China. We have expanded our webinar offerings over the course of last year. I'll note the first one at the top, which was a previous webinar called Getting Started with MedDRA. But because we want to expand the global reach and implementation of MedDRA, we are now opening this up to non-subscribers as well as subscribers. And this has been particularly welcomed in Latin America. So users can get a sneak preview of what MedDRA is, what it does, and how to subscribe. Also in green in the webinar column, we've had some very positive feedback about our Let's Code Together webinar, where MSSO coders discuss thorny coding issues and the audience was able to interact with them. This is a more advanced offering, but people really appreciate the interactive nature of these discussions. We also have the MedDRA version analysis tool demonstrations using practical examples, and an introduction to the MedDRA APIs, the application program interfaces for non-IT personnel. So I was able to benefit from this one as a non-technical person. So those are all interactive webinars where you can attend in real time, just as we're doing today, and they're also recorded. We also have instructional videos where you can view at your own leisure a number of how-tos for more technical aspects of using MedDRA, how to subscribe, how to submit a change request, and so on. But in terms of actual MedDRA training, we have an instructional video using more of an interactive format on structure and scope. And our most recent offering is interactive MedDRA coding. So after attending the training webinars for coding basics or advanced MedDRA coding, you can actually practice your skills and answer questions in an interactive course and get feedback on your answers. Also, we have just posted another introduction to MedDRA instructional video, again with interactivity on our website. So do check back frequently to be able to see what offerings we have. Now, a lot of offerings, but how do you navigate them? Well, we've provided a new training curriculum page, which really presents a roadmap to be able to master MedDRA. So it takes you right from getting ready, mastering the basics, coding, and on to data retrieval analysis. And it shows all of the various formats in which you can receive that training. So thank you for that overview. I'd like to now take any questions that you might have in a short time before passing on to the next speaker.
Okay, so no questions at this time, Judy.
Okay, great. Well, I will hand back to Martin to introduce our next speaker, Dr. Barbara Law.
Yes, please.
Do you want to take presenter yourself, Martin?
Yeah, I'm on it.
Great, great.
Can you see my screen?
Yes.
So, our next speaker then is Dr. Barbara Law from the Brighton Collaboration, and she will be talking about SPEAC COVID-19 adverse events of special interest list, the process and update. But just to introduce Dr. Barbara Law, she is a subspecialist in pediatric infectious disease. She has 20 years of academic experience as section head of pediatric infectious diseases at the Winnipeg Health Science Center and professor of pediatrics and child health, as well as medical microbiology at the University of Manitoba. From 2004 to 2015, she was chief of the vaccine safety at the Public Health Agency of Canada and has been an active participant in several global vaccine safety initiatives, including membership on the WHO Global Advisory Committee on Vaccine Safety, 2005 to 2011, and the WHO Science Working Group on Vaccine Pharmacovigilance, 2005 to 2012. In 2013, she was elected to the Science Board of the Brighton Collaboration with a focus on their setting standards, and that's a strategic, or that provides strategic priorities to help facilitate and promote common terminology and shared understanding of adverse events following immunizations, so the AEFIs she will talk about a lot in her talk. Barb took on chairing the Science Board in 2018 and continues to do so. She retired in June 2015, but has remained very, very active as a vaccine safety consultant and is currently involved in the safety platform of emerging vaccines, which is the SPEAC acronym, which is a global Brighton Collaboration project funded by the Coalition for Epidemic Preparedness and Innovation. And that is striving to have vaccine developed against diseases specifically impacting low and middle income countries like Lassa, chikungunya, Rift Valley fever, Napa, Middle East respiratory syndrome, and naturally since 2020, COVID-19. So welcome, Barb, and we are very thankful that you got out of bed very early because she's from the east coast of Canada, and it's quite early there. Barb, the floor is yours.
Thank you, Martin. Just let me get my screen set up here. My computer thinks it's early in the morning too for some reason. There we go. Can you see that?
I can see it.
Okay. Excellent. So thank you so much for that kind introduction, and it's a real pleasure to join the group and have a chance to speak about what we're up to in the Brighton Collaboration these days. And I feel a certain affinity because back in the day when I switched from essentially academia to the Public Health Agency of Canada, I had to come up to speed pretty quickly on vaccine safety and pharmacovigilance, and it quickly became a lifelong passion to work in the global community. So it's a joy to be here. And also I took MedDRA training, and I think that's when I first been up with Judy. So it's a bit like coming home. So I'm happy to be here and to be part of this webinar. So what I'm going to talk to you today is about a little bit of a brief overview of SEPI and Brighton and SPIAC. Then to talk about the work that we're doing, and especially to focus in on developing a potential list of adverse events of special interest that could follow COVID-19 vaccines. To talk about the process and format behind the ASI case definitions that are developed by Brighton, to look at our related resources and tools that are part of our project, and finally just a very brief introduction to the website. And there are several more slides that I won't have time to show, but that are part of the download and that will give more specific introduction or just examples of pages that are at the website. So first of all, the SEPI stands for the Coalition for Epidemic Preparedness Innovations. It's an NGO that essentially vets applications with the goal to develop, to a certain stage at least, phase 2, vaccines that target diseases of importance on the global scene, especially as identified by WHO. And Martin's already gone through the list, and you see them here, so I won't read them through again. What's relevant is that the developers that are working on these, there are many, many sponsors, each with their own approach. They range from small industry, startup companies through mega global pharmaceutical companies and academic researchers. And they have multiple vaccine platforms, everything from viral vectors using measles, as one example, or adenovirus, DNA, RNA, and so on. So you have many platforms, and that raises an issue around monitoring safety. And so Brighton Collaboration has had an interest in safety since 2000, or has been formed since 2000, with a goal to build trust in the safety of vaccines. And the primary way that is approached is through our mission, which is to develop internationally accepted standards for monitoring safety through the vaccine life cycle, and has put out many adverse event case definitions, guidance on collecting and reporting vaccine safety data, as well as templates that look at all aspects of vaccine safety for specific platforms. Just as an example of why the impetus that Brighton had to get going on this, when you look at something as simple as fever, one of the first studies that was done looked at many, many papers. I think there were over 40. Many were phase 2 or 3 clinical research trials, and they were just looking to see how was fever assessed and what was the threshold, where was fever taken, and what was the timing. And there were a huge variation, as you can see here, in threshold. And it became clear that there needed to be some kind of a standard definition so that we can all know what we're talking about when somebody says an individual had fever. So the SPIAC is a Brighton project, and we were put a proposal to SEPI and were funded in 2019. And we've been operating during the entire project under the umbrella of the Task Force for Global Health, which is situated in Georgia. And our aim is to support SEPI's goal of having high quality, standardized and harmonized evaluation of vaccine safety across their entire vaccine portfolio. So now I'll move in and focus more on COVID. And also to talk, this will give you an example of what the SPEAK project is all about. This focuses on COVID, but we're doing it eventually for all the SEPI target diseases. And we like to talk about our toolbox, which is the main area that I'm involved in. And again, the goals, as said, is to facilitate harmonized approaches to safety data collection and assessment, and to anticipate vaccine safety issues that could arise during clinical trials. We do that first part by defining the adverse events of special interest. We don't know that these would happen after a vaccine, but there's a potential that they could. And so we do landscape analyses and literature review, trying to determine what events could happen related to immunization in general, related to a specific vaccine platform based on what has been seen in the past, as well as how the wild type target disease presents. It could be something related to viral replication, which would be relevant to live vaccines, or to immunopathogenesis, which is essentially related to all vaccines. The second step then is, once we define the list of ASI, is to make sure that if there's no Brighton case definition yet published, to develop one, as well as to develop the tools, as mentioned, to facilitate a harmonized approach, and also to provide resources on risk factors, background rates in the population, and of interest to this group, codes, MedDRA codes, and ICD-9-10 codes that would relate to the ASI in particular. So the landscape analysis for the target disease, the process was fairly simple for well-established diseases like Lassa fever, Rift Valley fever, and so on. We would get 8 to 10 recent review articles, what we called primary references, and these were reviewed in depth, summarized, and each of 2 experts created independently an adverse event list and then sought consensus across them, also looked at secondary references of interest. But it was a much different situation for COVID-19, which as of last December 2019 began to emerge, and literally a tsunami of clinical reporting and evidence data, as I'm sure most are highly familiar with. So our initial list was developed in early February, and it was based on 5 reports, which was the first description out of China that went from initially 41 patients to 99 and upwards. Subsequently, daily screening of published reports in PubMed and the preprint articles through May 16th, and we developed, presented the list to the World Health Global Advisory Committee on Vaccine Safety, who adopted the list essentially. It was a good collaboration, and then we've continued to do ongoing systematic literature review updates, and we did update the list in December 2020. This just shows you that the various categories of events that we have put on the list. So this left-hand column is all about things that really relate to all or most vaccines. So the first 5 items, anaphylaxis, thrombocytopenia, generalized convulsion, these have been linked with almost every vaccine. GBS and acute disseminated encephalomyelitis, not frequently associated, to be proven to be associated, but frequently reported. They occur in the population, and because they're so severe when one occurs following immunization, it creates great concern, so we put those on the list. Then the events with proven or theoretical association, but associated with specific platforms, included things like aseptic meningitis and encephalitis, which are relevant to all live viral vaccines. Bell's palsy, and an example of that was the E. coli adjuvanted intranasal flu, which was licensed, I believe, in Switzerland briefly, and then removed from the market because of the facial nerve palsy that was associated with it. And a theoretical risk, which was seen with SARS and MERS animal model for vaccines, where the vaccine led to an enhanced disease. Then looking at the events associated with or after COVID-19, this has been fascinating and alarming to follow because it seemed like every week back from February through to May, there was something new that suddenly was emerging. So clearly ARDS, acute respiratory distress syndrome, was the main factor that was seen. But later on, the multisystem inflammatory syndrome, first in children, and now it's recognized this happens in adults. Many manifestations of acute cardiac injury, coagulation disorders, which is clearly important now, a loss of smell, which was not anticipated, I don't think, and then some odd skin-like lesions, skin rashes, organ injury, and so on. So this is the list as it stands right now. And in this next slide, I'll just show you where we are with our case definitions. So anything highlighted in yellow is already published. Everything on the left-hand column had been published. We've developed, as part of the SPEAK project and working with several of the consultants and led by Flor Munoz, the working groups have created definitions for ARDS, for the multi-inflammatory syndrome. We're nearly finished with the acute cardiac injury and coagulation disorders. And a working group has just formed and is actively working now on developing one for anosmia. And we will be going through developing additional case definitions, especially for the bleeding disorders and so on. So just to move now to the process and the format that's related with the case definitions themselves. It's one of the things that we have a standard process. We're supposed to be about standards, and so we have a standard developmental process for each and every Brighton Collaboration case definition. And it starts with a call for working group volunteers who generally, we want to be sure we have subject matters experts on the adverse event itself. And you can see we've had many neurology case definitions, hematology case definitions, and so on. So we need to have those experts to contribute. There also needs to be process experts who are familiar with the Brighton approach and can lead and support the working group and remind them what the case definitions, how they're meant to be constructed. And then it helps to have vaccinology experts who have experience with vaccine safety, epidemiology, and immunology. We also include, and part of the working group work is to do systematic searches to provide background to in the publication itself, or sometimes a separate publication on relevant epidemiology, prior case definitions, risk factors, and background rates in the population. And the case definitions are done and designed to accommodate variation in diagnostic certainty based on the information in an individual cases. So we talk a lot about levels of diagnostic certainty, and these go from definite cases, which is level 1, to level 2, which is probable, and level 3, which is possible. As you go from the lower levels up to the higher, you clearly increase specificity, whereas when you're going down to the lower levels, you increase sensitivity. There's always a level 4, which enables people to classify cases as it's been reported as such, but insufficient information to meet any of the levels of the case definition. And finally, there's not a case. And several of the case definitions, notably encephalitis or ADM, have specific exclusion criteria. So you can say this clearly is not a case because an exclusion criteria is met, which might be an alternative diagnosis for the condition. Anaphylaxis, as an example, you could have all the features, major and minor criteria of anaphylaxis, but if it isn't acute in onset and rapidly progressive, it's not a case. So here's the published case definition of thrombocytopenia. And just to use as an example, the goal of the case definitions is really to enable data comparability within and across diverse, very diverse vaccine safety settings. So I've had great interest in seeing the survey at the beginning, and the audience covers this spectrum. So from phase 1, 2, 3, and post-market clinical trials conducted by manufacturers, by researchers, to pharmacovigilance done in high, middle, and low income countries, and to epidemiologic research in order to develop background rates, as well as do causality hypothesis testing. The application of the case definitions, one application is to identify key info that's needed or missing in an AFI report. And we actually redesigned our national AFI report form in Canada to try to adapt to some of the more common and considered events of public health interest, like thrombocytopenia, as well as anaphylaxis and others. We enabled categories on the reporting form that would get the key criteria, so we would have that information when we were trying to classify a case if it was available. They can be used for epidemiologic case finding as well as case assessment for diagnostic certainty. And this is the first step now in the new revised, it's not new anymore, but about 10 years ago, the WHO causality paradigm. What the case definitions are not meant to do is to guide case management. And this is something that we needed to ensure public health authorities and the cases themselves are not constructed that way, nor are they meant to determine vaccine event causality. They're the first step, but once you've done that, there are other things that you need. And most of the, I'd say virtually all the case definitions refrain from giving any risk period following a vaccine when this could be a case, because they are not meant to look at causality. So there's over 66 case definitions published now. There was 25 focused on pregnancy outcomes, injection site reactions, 5 specific to smallpox vaccines, and then the last 29 are a variety of adverse events, many of which are on the adverse event list for COVID-19. They're all freely available for download at the Brighton website. So the goal of the case definitions, again, is data comparability. Oh, I'm sorry. So I wanted to show how the case definitions might link to MedDRA and especially to the SMQs. So this audience well knows the goal of SMQs is to aid in identification of potential cases. There's many formats, from narrow broad for things like Guillain-Barré syndrome, algorithmic for acute pancreatitis or anaphylaxis, and hierarchical, such as cardiac arrhythmias or the thrombotic events. The link, as I see it, between the two is having identified your cases, where it is important to confirm that a case is a case. You can use the Brighton case definition to assess a level of diagnostic certainty from definite through possible to not a case. And this is especially important when you're looking at signals. So the working group, as I mentioned, for thrombosis and thromboembolism in the SPEAK project has been hard at work for months, really, since the fall, and are nearly finished. There's actually one of their final working group calls is later this morning. And they've developed a case definition. It may get modified a little bit, but just to give you an example, because this is such a hot topic, the level of certainty 1 is a definitive case, and this is really a demonstration either by imaging or a procedure or pathology that shows there is indeed thrombus or thromboembolism, and it is completely devoid of any need for clinical findings or risk factors. Level 2, a probable case, is more a clinical presentation consistent with thrombosis or thromboembolic event, might include a specific clinical syndrome, or a non-specific one associated with a risk factor. And for both of the above, you would need supportive findings, but not definitive. So possibly chest X-ray, echocardiogram, and a lab test showing elevated D-dimer. And there should be no alternative etiology. The third level of certainty, possible case, is essentially the clinical presentation alone without any supporting laboratory information. This just shows an algorithmic approach, what we call an algorithm, which is really a decision tree where you can record your information and you can come to a diagnosis of the level of certainty. And in order just to show you the similarities, some ways, between the SMQ for embolic and thrombotic events, the relevant terms there cover the things in box A, such as the abnormal imaging studies or surgical procedures. It covers the box B, clinical procedures, clinical entities, thrombosis, embolism, and the syndromes, some signs and symptoms, and then the hemorrhagic laboratory terms, including the D-dimer elevation. And this shows our already published thrombocytopenia algorithm. We're currently working on a case finding definition for this new entity that's been seen following the AstraZeneca and some other of the adenovirus vector vaccines, and which includes thrombocytopenia and thrombosis. And it's an adaptation of this, which is currently underway. So just moving now to the ASI related resources and tools. We have developed, in association with the case definitions, companion guides, and these are all downloadable from the website. They include a lot of information, such as I mentioned earlier, the risk factors, the background rates, which is done by a systematic literature search, the codes relevant to the case definition. And then the next several tools are related to data abstraction and interpretation. They can be like a CRF, if you will, for use in medical record review, where it guides the searcher to get the information that's needed, the key criteria to meet the case definition. Suggestions for real-time investigation, and then algorithms like I showed you, the pictorial one, but there's also one that's more tabular in format that helps get to levels of diagnostic certainty. And these resources, again, are available at the Brighton website. There's also at the website a dashboard with background population-based incidents, which is more interactive, and an ABC tool similarly, where it walks you through for given entities. And these are the ones, the tier 1, we have the ABC tool is done for all of these, and the others are coming. And I thought I'd just show you for thrombocytopenia as an example of what we put in for the ICD-910 MedDRA codes, and some of the methodology. So the table here shows you, I'll just go through the methodology first. So this is based on technology developed in an advanced project called CodeMapper by Becker et al. And what it does is use the meta-thesaurus of Unified Medical Language System codes and terms from different source vocabularies, such as MedDRA. There's over 1 million concepts connected to codes from 201 vocabularies. And then the concept unique identifier, or CUI, links the UMLS codes with the same meaning. And each concept is assigned to one or more semantic types. And then there's this text indexing engine, Peregrine, which uses a dictionary to identify medical concepts in the Brighton case definition. And then we end up with something like you see here. And this is the table in the user guide, which we focus on MedDRA terms and ICD-910 that could potentially be expanded in the future. And you can see all of these in the manuals that we've completed from the website. So just moving to that, lastly, I'm not going to go into any detail on this, but this is our website page when you go to it, and you'll see that there's under pages, there's about midway down, there's a COVID-19. That's mostly what we seem to be putting up these days. There's also, you can go to if you look at posts trending today, the priority list of adverse events has a detailed document on how we'd actually developed these. And you can get all of the case definitions downloaded, as well as the companion guides. So with that, I'll thank you for your attention, and I'm happy to try to answer any questions if there are any. And I do hope you'll visit the website.
Thanks, Barb. Are there any questions?
Not at this time, Martin. So if questions do come, oh, wait a minute. Here we go. Let's see. Here we go. Question is, although a causal link has not definitively been established for VITT as a vaccine-induced immune thrombocytopenia, should the term be added to MedDRA, or would a term for immune thrombocytopenia be sufficient for coding and retrieval of cases? We realize that there are case definitions in progress.
Yeah, it's a question on everybody's mind, I think, and certainly it would be good to have a term for the immune thrombotic thrombocytopenia, and that's the case definition that we're working towards. Again, which will be actually a constricted one from what you saw. It combines thrombocytopenia and gets rid of the risk factors, because it may well be that vaccine itself is a risk factor, but we don't know that yet. So it may be that in the interim, having that term to capture the immune thrombotic thrombocytopenia, and then in time, and I would think it won't be long, because people are hard at work at this, and there's a lot of specific laboratory testing that can be done around the platelet 4 antibody to see if there are situations where you can clearly show causality, in which case then having a vaccine-induced thrombotic thrombocytopenia like there is one for heparin-induced may well make sense. I don't think we're there yet. Thank you for the question.
Okay, now, Martin, that's all the questions we have at this time, so we can probably move to the next.
Yes, so I'll grab the presentation again and share my screen. So thanks again, Barb, for this very, very interesting speech. And I hope many of you are clear that Brighton is doing a really great job in preparing actually the scientific background for all this SMQs and for your data retrieval and assessment concerning vaccines and vaccine safety. So thank you very much, and providing you huge resources for you to work with. So that said, we go into the use cases, and that is our next talk, which is from Anna Baumgarten from the Uppsala Monitoring Center. She will talk about MedDRA in global COVID-19 vaccine safety surveillance in the WHO program for international drug monitoring. And Anna is a Swedish pharmacist with a background of different roles and positions in community pharmacies in Sweden. She came to the Uppsala Monitoring Center 10 years ago to work on the development and maintenance of WHO drug. One of her focus areas at the UMC has been the medical terminologies used in the pharmacovigilance of the WHO program, and which is mostly MedDRA. But she has also worked with this precursor, WHOART, and mapping between the two terminologies. And during the last 3 years, her focus has been on pharmacovigilance, supporting national pharmacovigilance practices throughout the use of the UMC pharmacovigilance products. And she manages a talented team of pharmacovigilance officers in daily contact with the national pharmacovigilance centers of the world. So a very global position, and over to you, Anna, and thank you for being with us.
Thank you for having me. I'll just see if I can get my screen up now, right? I'm showing my screen, right? And then I'll stop sharing my picture. Okay, thank you for the introduction, and thank you for inviting me to this event. We live in challenging times, and we are all challenged by this pandemic to different degrees, of course, and I personally see myself as one of the fortunate ones. And I'm fortunate to work for an organization who every day try to make medicines and vaccines safer to use globally. So I'm happy to be part of the Uppsala Monitoring Center, and I'm here today to talk about how the UMC supports the COVID-19 vaccine safety surveillance in the WHO program for international drug monitoring, and how we are making use of MedDRA in this. But first, I would like us to take a step back, starting off with a reminder of what pharmacovigilance is all about. This is the origin of our field of work. Dr. McBride made an observation. He saw children being born with abnormalities when the mother had been given the drug thalidomide. He compared the frequencies of the abnormalities he saw with what he expected and concluded that it was an increase of frequency. So he posted this letter in the Lancet, where he asked, Have any of your readers seen similar abnormalities in babies delivered of women who have taken this drug during pregnancy? And we all know the story. As a response to this tragedy, the World Health Organization set up the HOPE program for international drug monitoring. The rationale for setting up this program was to make it possible to identify rare adverse drug reactions that could not be found through clinical trial programs. It quite soon became evident that maintaining an international database of case reports and a network of institutions and scientists concerned with drug safety issues provides additional gains when compared to operation in isolation. So this program has been a success and now covers 171 countries of the world. Countries that share the vision of safer and more effective use of medicines. They work nationally and collaborate internationally to monitor and identify the harm caused by medicines, to reduce the risks of patients and to establish worldwide pharmacovigilance practices, standards and systems. Sorry for having slightly wrong numbers here in the handouts though. We have welcomed 3 new members to this program during 2021 so far: Kuwait, Lebanon and Algeria. So the program's joint pharmacovigilance database, VIGBase, in which countries have been sharing reports of suspected adverse effects of medicines for decades now, continue to grow and now holds more than 25 million case reports, of which 500,000 reports are reports of suspected side effects of the COVID-19 vaccines. UMC have been responsible for the technical and operational aspects of this program since 1978, and we maintain VIGBase on behalf of the program. So it's basically UMC who makes sure VIGBase is up to date with the latest MedDRA version, etc. And we maintain and develop WHO-Drug to meet the needs of a global standardized drug dictionary. Not UMC, nor the WHO have any true regulatory authority, but we're actively involved in global harmonization efforts, and as such, we actively encourage the use of MedDRA in the program. Okay, that's it. Let's go into vaccine surveillance. As a response to the pandemic, WHO and experts from around the world got together and produced this COVID-19 Safety Surveillance Manual, where experiences from previous pandemics were combined with the expertise and previous manuals and guidelines for vaccine safety surveillance. And this is to guide setting up surveillance processes for the new COVID-19 vaccines. So what are the challenges? Many, of course, but one being the scale at which these vaccines are being rolled out. Millions of people receive these new vaccines every day. Even a very rare event, say affecting only 1 in 100,000, can potentially affect hundreds and even thousands of patients in just a week. So we need to act quickly. We need to do real-time monitoring of the data. And if we, instead of looking at the data just locally, regionally, or nationally, also do a global surveillance, we have the possibility of detecting even very rare effects quickly, given that the data is shared. So even if the COVID-19 vaccines questions are new, this is really addressing the same thing, isn't it? It's back to the beginning, where the vital question was, did someone else see this? And this is where we think that access to the WHO global database is really powerful, but because it can support you to answer that question. And looking into the manual that the WHO issued, we can read that this is exactly what it recommends. Countries are encouraged to review and adapt processes for timely reporting, reviewing, and data sharing nationally, regionally, and globally. To the members of the program, we offer the use of Visualize as a way to access the Vigi-based data, the global data. Visualize is a signal detection and management system that can use national, regional, or global data as both a reference and as a starting point for quantitative signal detection. It provides support for the entire signal management process, including the qualitative assessments. It is developed to support signal management and signal detection best practices, like the ones described in these documents, the Science Guide and the Scope Guide on this picture. In Visualize, the members, of course, can access the data in Vigibase, but the program can also use Visualize to share their findings, to share their signals. MedDRA is the terminology used in Visualize, and one can browse and search in MedDRA, make analysis and statistical calculations on all the grouping levels of MedDRA. Visualize is built to support the national pharmacovigilance processes, and these processes are to a large extent the same for drug and for vaccine safety monitoring. However, some aspects are a little challenging when it comes to the COVID-19 vaccines monitoring. One being the number of reports. Big number of reports can make case-by-case assessments very time-consuming, and one therefore might need to set up processes for prioritizing effort. Visualize supports this by providing easy-to-use statistical methods, for example, by providing measures of disproportionality for all levels and groupings of MedDRA, as well as being able to compare disproportionality over time or comparing national, regional, or global measures. This is already a common practice for many advanced pharmacovigilance settings, of course, but in Visualize, we can provide this to the entire WHO program. And we are very happy that the MSSO supports this by, for example, providing MedDRA training to the program. Another challenge being the number of vaccines introduced, approved under different names in different countries, and the different platforms they are being used, that they are being based on. So HUDRAG has come up with a solution to this, which we then have implemented in Visualize. So in Visualize, one can do the analysis, for example, by doing searches and providing statistical calculations for, say, all viral vector vaccines or all mRNA vaccines. Okay, that was Visualize, but in order to do the analysis in Visualize, the reports need to be there. In each country, data of suspected side effects of drugs and of vaccines are collected from different stakeholders. There could be paper forms, telephone numbers to call, pharma industry obligations to report to the regulator, apps for facilitated reporting like the MedSafety app Kendall will talk about in a little bit, electronic healthcare record systems, online reporting systems, etc. And to the members of the program, we offer support for this in Judy Flow, which is used as the national pharmacovigilance system and as the national pharmacovigilance database. The reports are then structured using the international standards. Whoops, what am I doing? Where MedDRA and WHODRAG are key components. And VigiFlow then offers controlled and secure sharing of information to VigiBase. And again, looking into the manual that the WHO issued, we can read that it recommends the use of VigiFlow for AFI reporting, as VigiFlow supports the processes described, from data capture to analysis. The purpose being transfer of adverse event following immunization data using the 25 core variables from the periphery of a healthcare system efficiently and effectively into a central database for processing and conversion into information that then can guide actions. This is to be able to provide quick and reliable information to decision makers at different levels in a country. The sharing of pharmacovigilance data is in many instances of the world standardized, but in many countries there are no established collaboration between national immunization programs and pharmacovigilance programs. Drugs and vaccines are monitored by different parts of the national vigilance systems, and sometimes these two programs have different focuses. This can, of course, be a challenge, but by using one system throughout the PV system, one facilitates not only transparency, but also speeds up the process, as there is no, or at least very little, need for transformation of data. VigiFlow is developed to support the national pharmacovigilance processes and is optimized for monitoring the safety of all drugs and vaccines on the national market. VigiFlow is built on the international standards, means that by using VigiFlow, one uses MedDRA. And as you can see on this map, VigiFlow, the blue countries, is used in quite a few countries of the world. Countries having different pharmacovigilance processes, ranges from countries starting up their PV activities to very advanced systems. And we are proud to be collaborating with the MSSO in providing MedDRA to these countries' pharmacovigilance activities around the globe. Almost not seen on this map, but we have a couple of European countries too using VigiFlow. We have recently developed increased support for vaccine safety surveillance in VigiFlow, developed by the UMC, but in tight collaboration with the World Health Organization. It applies to all vaccines, not just the COVID vaccines. And this is an entire new section of VigiFlow to capture adverse event following immunization based on the form WHO AFI reporting form. The WHO manual mentioned the use of MedDRA in this sharing of report of safety concern, but as we've designed VigiFlow to meet the needs in data capture, we've incorporated MedDRA in the very first data entry phase of the form. This means that here we are collaborating with the MSSO to introduce MedDRA to potentially new audiences, to national immunization programs in countries choosing to use VigiFlow for their vaccine surveillance. The ones in the immunization setting filling in the AFI form isn't necessarily trained in MedDRA. In some instances, the data is more of filling out tick boxes. Therefore, we were put with two options, either leaving the medical coding for the National Center, National Pharmacovigilance Program, to handle, or creating an easy-to-use subset of MedDRA. We went with the latter, as the expected demand for the pharmacovigilance program for the coding would be big if left uncoded. So we discussed with the MSSO on our way forward, and the WHO paper form identifies a number of commonly reported terms, and some more commonly reported terms were identified with the World Health Organization. The question was, can we make vaccine or immunization specific MedDRA coding out of these terms? As the context is already set by the form adverse events following immunization, we didn't want that information to go missing when we pooled this data with other pharmacovigilance data, nationally or globally. We also wanted these terms to be familiar with the terms that they were used to use in the paper form. So MSSO helped us do the MedDRA mapping. Commonly reported term in the vaccination context mapped to MedDRA. And this is how we've implemented it. When doing the manual data entry in Judy Flow, one has the option to report in free text or do MedDRA coding, but one also have the possibility to open up a list of commonly reported terms, and when one picks a term from the list, one is provided with the MedDRA mapping of this term. Of course, one has the option to revise the coding or to do several codings. Choose several terms. This has been well received so far, and having the AFI data, adverse event following immunization data, MedDRA coding from scratch really facilitates the rapid analysis. In the AFI reporting process, often the first step in the analysis is to produce a line listing, which is an overview of the data to support initial identification of clustering or any unusual or significant reporting events that needs further analysis. And the data is then recommended to be tabulated by place, person, time, antigen, type of event, etc., to get a first overview. And you all know the benefits of using MedDRA for this, as MedDRA can easily be presented in all the different term groupings, and it's standardized in all of this. So in this step of the analysis, it's possible then to identify common immunization errors, for example. For example, an increased number of abscesses by one immunization center could be more likely to be due to immunization-related error than the vaccine itself. Okay, that was how Judy Flow and Vigilize has been expanded to increase the support for vaccine vigilance in the program. But it doesn't stop there, does it? Going back to this picture, the Judy Flow targets to support the pharmacovigilance processes at the national regulatory authorities, but some of the modules connected to Judy Flow have applications for other stakeholders. As Judy Flow, it's based on the international pharmacovigilance standards, such as the ICH E2B. The standardization itself vouchers for interoperability with other systems. Taking the example of the MedSafety app, which Kendall will talk about very soon. This information from the app can then be made available to the National Pharmacovigilance Program for processing and analysis. And this collaboration is then really important to provide easy to use reporting channels. In many countries of the world, the DIS2 system is used in the national immunization programs. Not until the national immunization program can share their data with the national pharmacovigilance program, we can start talking about true benefits of the global approach to vaccine safety. And basically, as soon as the National Pharmacovigilance Center has data in the international standards, they can have it connected to Judy Flow, where we have MedDRA for structuring the information. But we all know that the industry and the market authorization holders play an important role in vaccine safety surveillance. evidence. The National Pharmacovigilance Center can choose to activate functionalities in VigiFlow to the industry, who can more easily fulfill their reporting obligations to the regulator. And we've been collaborating with the MSSO on this to ensure not only correct handling of MedDRA, but also to facilitate license checking, etc. And here we have development ongoing. Our plan was to have this fully launched already. But you all know what happened. A pandemic came in the way. We have an open access online reporting form for patients and healthcare providers as a connection to VigiFlow. And currently, we are working on expanding this to also being able to offer a separate vaccine-specific online reporting, e-reporting, based on the WHO AFI form. So to conclude, lots of work is ongoing, both in VigiFlow itself, but also in systems talking to VigiFlow to increase the support for vaccine vigilance in the program, making vaccines safer to use. And MedDRA plays a very important role in this. Okay, how about access to the global database if you're not part of the National Pharmacovigilance Program? All can access Vigi-based data, but it's just different level of access to the information due to it being sensitive data. And I put a slide here for your information on on different ways of accessing it, but you can read that yourself. I just wanted to wrap up by saying a couple of words on the signal detection UMC do in the COVID-19 vaccines in Vigi-base. So at the UMC, we do signal detection on the Vigi-base data. Our role is to complement the work of other organizations, exploring new approaches to signal detection and assessment, and looking for new types of signals, especially clinical patterns or syndromes or risk group factors. We will not replicate the work of large regulatory authorities and pharmaceutical companies who may have more timely and detailed individual case reports. It's also appropriate for us, for the program, to focus on helping or looking at signals for low and middle income countries and maybe preliminary vaccine signals that are not already being assessed by the bigger regulators. Signals produced by the UMC are made available to the program in Visualize and are often published in the WHO pharmaceutical newsletter. And in addition to our existing methodology, we have recently added a new approach based on collecting coherent reports, those reports that share groups of MedDRA preferred terms. By looking for patterns or syndromes, we believe that we can produce more insightful results than by simply looking for individual symptoms. And we're now implementing this new method to the COVID-19 vaccine signal detection, the aim being to identify clinical patterns as soon as possible, both expected and potentially unexpected. We are excited about this new method and hope to be able to share some results soon. Research findings such as method development are published in scientific literature and can often be found listed on our website. And I should also say, on request, we support WHO on their questions of safety concerns on the new vaccines too, of course. Thank you for listening. So let's see, do you have any questions? And
yes,
let's see if I can respond to that.
We have 3 questions. First question is information on narratives available from Visualize for pharmacovigilance centers?
Information on the narratives?
Yes, information on narratives, is it available from Vigilize for pharmacovigilance centers?
Yes and no. As narratives could or does include information that could be regarded as sensitive. This is the property of the National Pharmacovigilance Program, the National Pharmacovigilance Center. So they are not generally shared to others. There are some instances where this information can be accessed by people outside of the own national program, and that would be for WHO to do their in-depth analysis, or the signal detection team in UMC to do their in-depth analysis. But it's not generally shared as it's regarded as sensitive information. Was that a response?
Right. So next question, very quickly, is, you may have answered this in your presentation, but what is the rationale for only providing Vigilize access to national pharmacovigilance centers? Why can't it be public?
Oh, we would love to do that. It is the property of the program, and so I would say if the access levels were so that we could. The nature of the data is, of course, sensitive. If we have any way of solving that so that we can make the data available without being, when being anonymized, then I think we can solve that. And of course, that is something that we are looking into solving. But for now, no, we can't access.
All right, and the last question is, I guess this is asking you to make a guess here. What percentage of all vaccine reactions do you believe are being reported to the UMC?
Very, 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 very little. Some programs we see, they report everything in a very timely manner, whereas other members of the program have either have maybe technical issues between the national immunization programs and the pharmacovigilance programs, or don't have established processes for sharing the information. So I don't even dare to guess. Someone more into the vaccine field needs to do that guess, I think.
Okay. Thank you. And then we just got one last question in here before we move on. And I'm going to send this to the chat because it's using a lot of acronyms. So if you can look in the chat, but it's asking, will the AEFI predefined list be expanded to include other AESIs such as GBS? So if you look in the chat there, the question is provided.
Yes, no, this predefined list that we've worked with is basically a list that the WHO has provided us with. If we see that there is an interest in increasing this list, absolutely, we can add GBS too.
Okay, thank you. That's all the questions we have. So, Martin, I'll turn it back over to you.
Yes, thank you. And I'll try to share again. Thank you, Anna, for this very interesting talk and concerning the general adverse events of special interest. You've heard Barb and the list there. I think that already includes GBS, just for that concrete question. And there is an SMQ specifically for GBS available, Guillain-Barré syndrome, for those who don't know this acronym. So that said, yes, already we have already Kendall sharing her screen. This is Kendall Harrison. Kendall Harrison is the Pandemic Systems Delivery Manager in the Vigilance and Risk management of the medicines divisions at the UK Medicines Regulatory Agency, the MHRA. She has worked in the division for 10 years and previously been responsible for the handling and assessing reports of suspected side effects as well as signal detection activities. And more recently, she has led work streams for the WebRadar 2 project, which has a number of deliverables, including provision of mobile phone apps to low and middle income countries in a joint collaboration with the WHO. This work seeks to enhance reporting of suspected side effects internationally, as well as to deliver information about medicines to app users. Using the same WebRadar infrastructure, she is now leading on development of pandemic systems to ensure that safety information is collected for products used in the pandemic. So that said, thank you very much, Kendall, for talking here, and the floor is yours.
Thanks very much, Martin. So I hope you can see my slides and hear me clearly. Do shout if not. So thanks for that introduction. As Martin said, so I'm Pandemic Systems Delivery Manager, so much of my talk will be talking about the systems we're using and enhancing to support pharmacovigilance and detection during the pandemic. And for those of you who are regular attendees at these sessions, I gave a talk a few months ago to the MedDRA user groups about our systems, and since then vaccines have come in. So I'll give you a bit of background or further information about what we've been doing specifically in the vaccination scene. So we'll talk about the extension to the systems that we have specifically for vaccinations, because the systems we have were originally developed back in and made public back in May in quite a rapid turnaround to get them up to speed to collect information about medicines that were starting to be used off-label for treatments, as well as for device products such as PPE. But there was always the intention that this would be extended to vaccines in due course, which of course it now is. We've also done a lot of work with our WebRadar infrastructure, and as Anna mentioned, our MedSafety app. So I'll quickly run through what we've done to support other countries with the use of MedSafety app and how that nicely fits in with the Judy base and Judy Flow aspects that Anna's just talked about. I'll then talk briefly about our vaccination strategy and the MHRA publications we're producing. This is all publicly available, so I'll talk about it at a very high level, but there's lots of information on our website if you want to go into that thereafter. So this is the website that we have. We, as I said, surfaced this back in May last year, and as of December we launched vaccinations within the UK, and the first reports of vaccinations started to come in. As we've started rollout to new vaccinations, those have been added. So most recently, the Moderna vaccination has started to be used within the UK. So we now have Pfizer, AstraZeneca, and Moderna vaccinations being used. And of course, if further vaccinations are available, then these will also be extended. The kinds of questions that we've asked for vaccinations have been enhanced as well, such that they can ask specific questions about vaccinations. We know the standards for collecting vaccination information, in fact for biologicals more generally, requires that we ask for batch numbers, and so as well as brands. So what we have done is we support the user by auto-populating the vaccinations, and then we also prompt the person to add batch details so that they can ensure that information is collected. We've making sure that where possible people are being told about their batch number, so the reporting of that is high. We've also specifically asking about first and second doses, a lot of important information about if people have reactions differently to first or second dose. So that's been added as an additional question that people can populate. And of course, we also ask for dates, which is important. We were also aware at the beginning that we wanted to collect information about the patient's immunocompromised status. So we added some specific solicited questions really about particular circumstances that might impact a person's immune system. So we have a drop-down list for which people can add. Essentially, it's helping add that patient medical history, but in a more structured way. And of course, all of these map into MedDRA terms so that it makes it easy for us to analyze reports based on the patient's status. We've also asked questions about the patient, whether they've had a COVID vaccination infection post-vaccination. This for us is a remit that actually falls outside of the MHRA, but it's of interest for us to understand where patients are having COVID-19 post-vaccination, not least because we can then see whether it's within the first few weeks where you might think that it takes a little while for the vaccination to become effective. So we do ask questions about that and the outcome of the infection. And just to show that whilst the image I showed you was our website, we also have an app, and this is what's built through the WebRadar infrastructure, which I've talked about previously. The questions that we ask on the app are identical to the website, but they just look slightly different. We talked about this in the last session, but just to recap, we also asked questions about COVID status prior to vaccination, and another area of interest to understand whether people with prior COVID infection handle vaccinations or have different types of or severity of reactions to people who've never had a COVID infection. So this has been available, in fact, for medicines, and it's being used as well for the vaccination type reports, and all of these map into MedDRA terms automatically onto our database such that it supports easy analysis of that and collection of that data. And we've not just been working on UK surveillance, we've also been supporting the MedSafety app. And just to recap, the MedSafety app is an offering that we work alongside WHO and UMC to offer to low middle income countries typically. We have built this up using something called WebRadar technology, which is a project that was developed and run over the last 5 years or so to support new technologies. MedSafety app is now used by over a dozen countries to support pharmacovigilance surveillance in their own regions. And on the front is a picture of how you would load that. Importantly, one of the offerings which is important for the vaccination rollout is that people can report offline and then send the reports as and when internet connection is established. So for vaccination, particularly in rural locations, this is a really important aspect of the pharmacovigilance in those countries to get real-time data on those vaccination rollouts. We have previously done some work to import COVID-specific questions to it, and you can see on these slides really how that looks across different med safety countries. So each country looks slightly different based on their own branding and so on. This is translated into different languages, but fundamentally it's the same questions, the same setup. Countries are able to tailor them should they wish to, but the baseline is essentially asking the same questions, which helps ensure that everybody's getting the benefit of the enhancements that we've been offering. What we've spent a lot of time in the last few months is supporting the AFI form. So in the same way as we saw in Anna's slides, we are much more familiar, or certainly vaccination systems and sites are more familiar using paper forms. Because of the volumes and the fact that obviously technology has improved, it's important that we are able to harness the technologies to support AFI reporting from vaccination centres. So we've done a similar piece of work to UMC where we've been mapping the paper forms into the app system such that people can start to use the app as opposed to the paper forms, which has a number of improvements or benefits such that reports can get to the safety regulator in real time, and the quality of the data can be higher because you have validations, and of course it's also mapped to E2B, which is really important. So we spent some time and did some collaboration with UMC on this to make sure that there's consistency in the way we map, such that they do map into Judy Flow appropriately. And you can see on the right hand side we've now had an extension, so there's a reporting option for adverse events following immunization. And the way in which the forms have been enhanced to enable this is some of the questions have been added to make the questions identical really to the paper form. So on the left hand side we've got the questions relating to your vaccination. So we ask about the product. We limit the vaccinations to select from to be the same as what the country is rolling out, so that keep the quality of information high. Vaccination dates, first and second doses, batch and lot numbers, and the expiry dates. We're also doing a piece of work at the moment to enable diluent information to be collected as well. So there'll be some further enhancements going in over the next few weeks. The solicited reactions that are expected or most likely to be reported have been added as checkboxes, so people can still look at or report against those familiar reactions. These are all mapped to MedDRA terms. A few of them, such as the severe local reactions, have been mapped to a similar MedDRA term, but the quality or the content is always mapped to the clinical narrative, so the granularity of that is never lost. We also, in the same way as Anna mentioned, we have, in addition to the reaction pickers, the ability for people to put in any reaction they want, so they're not limited to that list that's available to them. In line with the paper forms, we also ask for the dates, the onset time, so how soon after the vaccination did the symptoms occur, and outcomes of that. And this has been successfully deployed to 3 countries so far, with ambitions to roll that out and enable other countries to take on that. See, each country is at a different point in their vaccination rollout, so we're in a good place to be able to support those countries when their pharmacovigilance requirements are there. And just bringing it back now to the UK strategy for surveillance of vaccinations. It's all available online. The link there is a bit long, but if you were to Google COVID and MHRA and gov.uk, you'll be able to find this. So we have 4 parts to our surveillance strategy. We have the enhanced passive surveillance and observed versus expected analysis. So this stems from our normal spontaneous reporting system, so the yellow card website, which I just showed you through, making sure that we are analysing reports of suspected side effects. So really for us, that's the bread and butter that we're also familiar with, but making sure that, of course, those observed versus expected analyses are done to put things into context, which is it's a challenge given the volume of the rollout and so on. The next is our rapid cycle analysis and ecological analysis. We are lucky to have linked data through a system called CPRD in the UK, which allows us to look at large population based data and do epidemiological analysis of reports and to see what's happening and what's put into GP clinical systems or hospital systems. The third one is a targeted active monitoring called the Yellow Card Vaccine Monitor, which I will go into in a bit more detail in just a moment. And then finally, we have the ability to do formal epidemiological studies. So this is an opportunity where there's a specific research question that we can do further studies to look into something in a bit more detail. So Yellow Card Vaccine Monitor is a system or a platform which was requested by our Commission on Human Medicines to be able to proactively follow up with patients after they've had their vaccination. And importantly, this differs from our spontaneous systems because it means that we will follow up with people, even those who have not had a side effect. Spontaneous systems often have a limitation where it's hard to put numbers into context because often the number of people having a drug or vaccination can't be completely tied together to the amount of reports you're receiving, whereas the vaccine monitor allows you to put that into context. It also allows us to follow up with patients over a period of time. So if we follow up with them over time, so often again, spontaneous data has a limitation where often you get data at a specific point in time. Whereas this allows us to get that more longitudinal data set, which is important. The process for the vaccine monitor from a patient perspective is that in the UK we're inviting people to join the vaccination monitor via our call-in system. So for those who are invited for vaccination, a subset of those people are being asked to join the vaccination monitor system. We ask the patient then to read the details and agree to participate, for which if they don't want to, they will not be able to continue. They then register to provide all of their background details, including past medical history and background, and also the date of their first vaccination, when that will be. at the point of their vaccination, we then contact them and ask them whether they did indeed have their vaccination, and if they have, how they're feeling, if they've had any side effects. And then we go on to follow up with them over the course of around about a year, to essentially follow up with them quite soon after their first dose, so 7 days after their first dose and 14 days. We will then continue to check in on them on their second dose and when that's due, and again quite quick follow-ups thereafter. And then we go into slightly longer follow-ups where we check in after about 3 months, 6 months, 9 months and 12 months to check how they are and if any of the information they previously provided has changed. Also allows people the opportunity, if they'd had ongoing side effects at the time of reporting, to be able to tell us whether those are now resolved. So it's an important part of our surveillance activities, which we are currently in the assessment process for. And what we're also doing on a regular basis is publishing the data that's been produced by our spontaneous systems and any analysis that's been done. So again, the link is at the bottom. You'll be able to find that and read up in more detail. But fundamentally, MedDRA plays an important role in the data that we're providing. You can see here one of the assessments that was taken from an earlier version of the report. Talks about specific assessments based on using MedDRA terminology. We also provide analysis prints of the reports that we've received, and importantly, these are all displayed in MedDRA terminology. So from system organ class 2 down to the HLTs and PTs, so all of that information is provided in MedDRA, and without that, that would be a really difficult part of our surveillance to be able to portray to people. So really taking advantage of MedDRA in that way. So please feel free to go on and have a look. There's more detail in the PDFs, and as I say, we are publishing those regularly. So I'll stop there given the time. We've not got many minutes left, but I'll stop there, and if there are any questions, I'll be happy to take them now.
Okay, terrific. We have 3 questions. The first one is, what is the format of these generated reports? Is it E2B? And are these reports sent to MHRA and then forwarded to the Pharmacovigilance Center, or are they directly sent to the appropriate Pharmacovigilance Center?
So the format is E2B. We currently have capability for R2 or R3, but practically most are in R2 at the moment. And these cases are sent directly to Judy Flow. We have systems, I guess, like an intermediary system, something called our Vigilance Hub. So from the Med Safety, the cases are sent to our Vigilance Hub, which is like a staging area where the countries are able to have access and look at the reports, but they go directly onto Judy Flow. So all of that data is aligned.
Okay, thank you. Next question is, who determines the seriousness of the reported events, since all events are reported as serious? What is the reason behind this approach of making all events as serious?
So, not sure if that's entirely true, but we basically, I mean, through SIOMS, we ask every reporter to decide whether they think their report is serious, and we mandate that. So actually, even if you're a patient or a healthcare professional, we ask you whether you think the report you're providing is serious or not. So they have the choice to define that based on their opinion. But we, of course, also do assessment both within the MHRA and our independent advisory panels to determine seriousness or assessment of particular reactions.
All right, thank you. So the last question we have is the public aware of coding, for example, using the general term tenderness instead of injection site tenderness or muscle tenderness, which they are under different SOCs?
I suspect the answer is that their knowledge is limited. We do have the MedDRA is available within both the web and the app versions of our system, so they have a look up against that, which is a contains field. So if you put tenderness, you've got the option. But we also look into patient narrative or the narrative text and so on. So there is, if somebody said tenderness, but actually it's injection site tenderness, then we do review those to make sure that it's in the appropriate SOCs. but yes, I suspect patient understanding or awareness of that is fairly limited. So we have to make sure that we, as a regulator, are aware of that and doing that recoding where appropriate.
Okay. All right, 2 more questions have come in. This first of these 2 are all vaccines followed up for through the yellow card vaccine monitoring?
Through the vaccine monitor, we are doing automated follow-up at certain times. So yes, all of the vaccine monitor cases are being followed up. And for spontaneous cases, we triage them to follow up the most important ones. Sometimes the information we provide is fully comprehensive, so there's no need to follow up. So we have review processes in place to make sure that they are triaged and followed up where appropriate.
Okay, and the last question I have is, who determines causality of each report?
So the assessment, we do a signal test assessment on all the reports, and that step we do MHRA, and it's also done through our independent advisory groups. So for that causality, we do it on a signal level rather than on a specific case level, but that's done at different stages throughout the process.
Okay. And then we just received another question. How many people at MHRA are involved in COVID-19 vaccine safety monitoring?
Oh, I don't actually know. I mean, the agency's 1,300 and lots of people are involved in different aspects of it. So I think that'd be quite a difficult one. I suspect nobody's not involved to some extent. So yeah, I'm not entirely sure what the specific figure is, but obviously it's been prioritized as an important thing that obviously lots of people are involved on to support.
Okay, and then another question here. How many submissions, reports, users do you have from the app?
From the app specifically, I don't have that to hand, I'm afraid. The publications that we have will tell you how many reports we've had totally for COVID vaccinations across the website and the app. So if you're interested in those volumes, then that's all publicly available for you. I don't know the app volumes. We certainly get most of our reports through the website still, but we do see good numbers coming through the app as well, and increasingly so.
Okay, well, that's all questions we have for this presentation. Martin, let me turn it over to you for the open question. We do have a go back while we transition to the open question period.
Yes, please.
Okay.
I think we can open the open question section.
Okay. So let me go back here. I believe this question came in as we were transitioning away from Barb Law's presentation. So I assume this was for her. Let me just get the question very quickly. Okay.
I have it. Excuse me. I
have it there,
Brian. Yes.
Okay.
So yes, I think this question is directed towards Barb. Were there any adenovirus platforms that caused thrombocytopenia?
Yeah, actually, I was checking the questions in the chat and I saw that one. I mean, aside from what we're seeing right now with the AstraZeneca and the Johnson & Johnson or Janssen, we are seeing, but it's not yet causally associated. Brighton, actually, I went to what was published on the vector templates, and there was one on, I think it was the AD26 vectors, and this is downloadable at the website. It's by Koosters et al. And on AD26 viral vectors, I searched the article for thrombocytopenia and it doesn't come in. This basically summarizes all the information that there is. So that would be a no for that one. And it seems to be the only adenoviral vector one that they've looked at. And we've had some discussion. I'm not aware of anything that's come up before this, but that's not definitive. I'd want to search it out. Over.
Okay, so far that is all the questions, open questions we have. So if we wanted to give it another minute for people to send in any questions for the panel.
Yes, yes, and definitely I want to also thank everyone. I want to thank our presenters. So thank you very much, Judy, Barb, Anna, and Kendall. So in the direction where they appeared here. So thank you very much for your presentations here and your participation in all the work you did. A general thank you to all of the MSSO to support these exercises here and to provide the platform. Liz, Brian, for you to help directly with the technical support, but to the whole team of the MSSO. And also to my fellow people within the steering committee, because there's a lot of work behind the scenes, organizing things and talking about things through. So thank you very much. But also to you as listeners to participate, to ask your questions, and we all hope that you will stay tuned in the future. But please, these is potentially one or two questions that could be placed, and do so as soon as possible.
Okay, I don't see any questions coming in, Martin.
So if you don't have any questions, I've already said a big thank you to everyone. I hope we will meet again. I am very hopeful that potentially early next year we may be able to have a face-to-face meeting again. We will see. We will be looking forward very much to see you again live and somewhere within Europe. But again, a very special thanks to Barb due to having to get up that early. It was 6 o'clock in the morning for her, so that everybody knows about it. And yes, thanks again. And look also into the additional information provided in the PDFs. There's huge of information also on the website of Brighton, of the MHRA, and of the UMC and WHO. So thank you very much, and I wish you a great rest of the day wherever you are. Thank you very much.